Hello, everyone. Yeah, I am Daqing Piao in the School of Electrical and Computer Engineering in Oklahoma State University, and I'm delighted to present some ideas that I have on the biophoton, and the topic is spatial and the temporal coherences of a biophoton emission, and what if they have a connection to consciousness. So biophoton is a pretty complex topic, and I only want to project something and for biophotons of humans. And I think it started with so-called ultra-weak photon emission of live organisms, discovered by Alexander Gurevich. And it has some very special properties, as we know of now. First, it's very weak. So you need either the very highly sensitive P photomultiplier tube or a very highly cooled CCD detector to detect them. And if it's about maybe 3 orders of magnitude weaker than what our eyes can sense. And about peculiar properties that it's also pretty broad band. I guess there may be more on the ultraviolet, but there are indications that they could cover entire visible spectral range. And it's spontaneous. So, but it's also shown that it's inducible, and particularly from the living organisms that can be stressed. So there are some nice patterns that you can see that it's related to the change, the variation of biophotons relates to the stress level, and its counting statistics. It's also just not like other photon emissions. So it's something that is strange. And early on, it had a lot of names tied to it, but I guess Professor Popp just coined the name of biophotons, and I'm particularly interested in biophotons of humans. So there are quite a number of literatures that have shown some very conspicuous and mysterious but weak patterns of biophotons of humans. And I think they need some kind of explanation. There are some, but there are a lot of stuff that are needing to be explained. One thing is the spatial pattern. That means it's acquired from whole body, from a healthy people, and a group in Japan is particularly active on that. And you can see this biophoton emissions from healthy human subject. Looks like the head and then shoulder, this area, it have more the biophoton activities. So this is a spatial pattern, and there is also quite several European groups, and one group, they also basically combined the measurements from human beings, and then they showed this kind of spatial pattern of biophoton that is more or less consistent. If lower at one side, it will maybe lower at the other side. So something like this, and according to this paper, that the concept of a pattern was further studied by focusing on the emission gradient from low emission sites over the abdomen to high emission sites over the forehead and the palms of the head. And essentially, they referred to that as a common human anatomic percentage distribution emission pattern, which means there are some very consistent patterns from healthy human being of this biophoton. And this spatial pattern of biophotons, they also show correlation, interesting correlations. You can call them as coherence. For example, this earlier work, you see that at the baseline, the right hand, the left hand, they are biophotons. This looks like they have similar phases, but the one from forehead, it's shifted in phase a little bit when the human being is basically stressed with light, and then you can see a significant change of biophotons. And then this is the delayed luminescence, the right hand, left hand, they are still quite much in sync with each other. The forehead had some phase shift. So there is this kind of spatial patterns that are correlated with each other. You might end up with some phase shift, but they are more or less correlated. And here is another one that from the group, they showed some changes of the biophotons associated with some disease conditions. And this sclerosis case, I don't know, it didn't mention which hand had the problem. But another study from Korea showed that for the patient with hemiparesis, the one that biophoton is stronger at the normal side between the two hands. And so this is left hemiparesis, this is right hemiparesis, and you can see the higher biophoton emission from the more normal side. So these are the spatial patterns associated with pathology. So those are interesting, difficult to study, difficult to duplicate, but are very interesting. On the other hand, there are also some temporal patterns that are quite consistent, and it has been repeated by multiple groups. And again, this is the Japan group. They show that this kind of the change of patterns in the morning, in the afternoon, it varies. So it's kind of hours to day long patterns, diurnal patterns. And here is has been observed over a few days, and they saw that this increase of the biophoton emission over a few days negatively correlates with the reduction of cortisol concentration in saliva. And there are also another study that to show that this kind of variation over about a day or so. And this is apparently it's some kind of sinusoidal changes overlapped with some variations. So something like this is very interesting. And I also see this study as very, very interesting, but this one has spent much longer years. It's a temporal pattern observed, and then it's from a 2016 paper, and that's spontaneous biophoton emission measured at the acupuncture point of LI1. It's at the tip of this index finger. And they basically measured over a large number of people. And you can see this from about 10 years old all the way to 75 years old. And this photon counts, again, for calibrating, normalizing this would be a daunting task. But if this study was very reliable, then we can see that an increase of this biophoton emission from this acupuncture point. And I would argue that, okay, so that it was for male, that was for female. So this part might be related to menopause. And this apparently would be the starting of the puberty. So this kind of patterns are quite interesting. But more interesting to this community might be the biophoton variations associated with the consciousness or mind state. And there are some studies we know of from the Wijk, and then for the biophoton changes, very conspicuous changes during the healing session. And so how to explain this? And also in Japan, there were many studies, count quite a lot of studies from this IS list, and to indicate that biophoton changes as a function of mental states, concentration, meditation, the changes varies a lot. And also I know that Dr. Cruz's Rhine Research Center also does this study and is measuring the biophotons from energy healers, which means there are some connections, but will meditation increase the biophoton, or will another altered state of consciousness reduce biophoton? So these are still not known. So over here, so we have essentially a matter that is a biophoton that can be measured. It has a spatial distribution, it has a temporal variation, it has some kind of mind state related alteration. And if so, the theme of this conference is connection. So I'm trying to make some connection with the mind. So and this mind, as we know, we'll talk about consciousness, but it's just too big a topic. But the consciousness will boil down to the state of mind. The state of the mind will boil down to how neurons fire, how they connected eventually. So that's, but you can't, I don't believe that the mind can be isolated from the neuronal firing or neural connections. So eventually it might be some kind of autonomic modulation, and this could indicate physiological impact, pathological changes, or some kind of psychology factors. So we have this matter, we have this mind, and then how are they possibly connected? So that's what I'm very interested to find out. So what do we know about the biophoton? So we have this biophoton over here, which is a light emission. And then I think it's almost established that this one got related to mitochondria when during the organism life cell, when it just take oxygen in and out, there is oxygen consumption. So this kind of excited states. And there was a nice review paper by European groups, so they put this together. So we know that this happens this way, although this is probably not a done deal, but this explains most of the stuff. So it's a kind of a multiple excited stage transition. It has this, so it causes this some kind of a broad band spectrum. But how would our consciousness possibly play a role over here? So that was the question that I would like to pursue. And in my two papers published in last year and a series of papers, I was trying to basically try to understand the biophoton emission from this decay, the stress response perspective. So I consider that, okay, this biophoton is detected, and then but this biophoton come from somewhere. You have to have somewhere to generate the mitochondrial activity, to generate the photons. Then the mitochondria, the respiration oxidative activities might be controlled, affected by external stress, but in human cases, it might be controlled somehow by some autonomic neuronal activity. So I consider this as, so any change, let's assume we have a consistent mitochondrial activity, consistent, stabilized, steady state of the biophoton, and then any change of that, it has to come from somewhere. It might come from exogenous stress, but it might also come from our brain does something to alter that. So with that, and then I put some, basically I consider this is the photon detected, and then it's got to start from somewhere within the tissue or organism, live organism, and any change of that got to come from somewhere. So there is going to have some kind of a transfer pathway to transfer the stress to the final photon detection. So I put out some models and try to model there eventually to get that hyperbolic pattern, while you don't have to have that coherence whatsoever, some kind of those Poisson. It doesn't have to be the hyperbolic pattern to reach this shape. It can be something else. So, but with that, and so you can have different stresses like photic, chemical, and different stresses. But what about if this change of the photon generation to be measured as a variation of a biophoton? Actually, what if that comes from our neuronal modulation? What if our consciousness can change that? So if that's the case, and then if we know what's going on there, and then there is a pathway, then we can correlate with the measurement. So, but in order to do that, we have to have some currency, as Jane and Dunn and in there, the subjectivity of science, they mentioned you just have to mint your own coins. So I tried to do something like that by doing this. So here, so I came up with something. So first, so this basically tells you that when we are at birth, all our neurons are fully intact. We have 100% intact neurons. When I'm dead, my neurons are died, 0%. So this is objective from 0 to 1. And also, let's assume all my 10 billion neurons, they all do things in complete synchronicity. Basically, they're all coherent, then I'm going to reach a maximum coherence. But if my brain, my neurons are completely out of phase, they are just completely random, then I'm going to be at this 0 state. So I call this a degree of neuronal synchronization or neuronal coherence. Here is another one. And then those are objective measure. And then I also somehow basically all the neuron firing spectrum can be put together as some kind of energy density. So you put them all together, it's an objective energy density term. I came up with I call it a vivo energy, which is actually energy density, joule per cubic centimeter stuff. But the reason I'm putting this is that if this one is related to our neuronal activity, and then when at birth, it's essentially a measure of how ordered our neuronal activity is. So when I was just born, I should have been in that optimum condition. But as people grow, it's going to be modulated by this physiology, which is a systematic change. And then if I got, my leg doesn't work well, which means I have some pathological factors moving in. And then, but if in the hemiparesis cases, which means your neuron will be able to control certain part of the body, but not the other part of the body. So basically over here, this is the birth state. That is physiology, systematic, that is pathology, and this is neuronal control. So I'm putting them all together, but I came up with a very nice article to tell this, basically this is essentially a life expectancy. So maybe this is a good way to describe how this combined, I call it vivo energy density changes over this lifetime. At birth, it's this much. But as we grow, we're just going to get lower and lower. When we get to this 0, that means we're done. But this change can be modulated by systematic physiology, just a normal metabolism. So it will basically oscillate like that with respect to this change. But if there is pathological conditions, then it's going to further just lower this one. And this one gets 0 earlier, which means people die earlier. So it's just kind of a quantitative way, try to give me a hand to push up. But I consider that change to cause metabolic change. So here is basically a physiological change of that energy state. It's a pathological change of energy state. And then I introduced the so-called physiological transfer factor, pathological transfer factor. The reason is to make this a conversion such that I have a light intensity to work with. So here I have a systematic light, I have a pathological light, and the systematic light is holistic, so I consider that as, just to make it simple, I cannot work on something like that. Just starting with simple cases, the biophoton light source in the middle of my body, and if it's pathological, it could be anywhere in the body. So, but by doing this, then have to do something with standard the engineering terms and with boundary conditions, how light propagates in tissue. But with all the things taken care of, actually I was able to calculate. There is a very small, in this case, it's a normal human model, which means this pathology. This term is 0, so a healthy source over here to emit the biophotons. And then, so this one is associated with only 0.01 microwatts of source right in the middle, about this big, bigger than my brain. And then the light intensity that you can measure at the surface is actually 10 to the negative 14 watts per centimeter square, as actually it's consistent with biophoton measured from human surface. So try to get to there. But then, so when this, I'm not inferring that this is aura, but I'm just saying that something like this can be interpreted as some aura-like hues that will cover the human being. So I don't know what aura is, but if this is one presentation of a bioenergy, and then there is a way to try to quantitatively model that. And then let's assume this is a healthy person under just a normal physiological the physiology, and then but the environmental change will cause will just through our autonomic neuronal control or sympathetic control whatsoever, it's going to change this metabolism. So this metabolism, let's assume that this source just oscillates a little bit over a day. And then, so if this oscillates, this surface measured one, it's going to oscillate. So it's going to get something. So I was able to just by also change this source a little bit, I just can get this. Apparently, if you add more variations, you're going to get something like that. So mimic that one. But on the other hand, so what if you have a much longer change, for example, over 70 years of change? And you can see in my model, I consider the change of this, my vivo energy term, the change cause the transfer to this one, which means the higher of this value doesn't mean stronger of the source. It's the bigger of the change causes stronger of the emission. So toward this, it's slow. The slope goes deeper, which means you're going to see a stronger change. So by using the gradient, and then over this years, I was able to just mimic this change. And then the women, they have more fat. So even though you have the same light, you get less at the surface, or they may be lighter, so they have a less total volume just by tweaking of those. And I can mimic this one, but but I didn't include that part. Certainly, for a model in itself should be able to make it something like that. So, but the one thing that I would like to specially indicate is that because I'm using a gradient, that means using a change to convert that to biophoton, which means let's assume at the very end of the life. Even though this amplitude is very small, but you're talking about a sudden stop of that little whatever energy state you want to cause it. That's going to cause a very big change. And this very big change could, in principle, give you a very strong sudden burst of a biophoton, which means it potentially can explain some anecdotal, the burst of flare or flash observed by the end of the life. So I'm just trying to through this possibility and just for people to think about whether this is possible or not. On the other hand, here is another one. There was a study by mice model of under human breast cancer. And they showed that, so here is a mice and the tumor at the right side, and as the tumor grows and then biophoton emission increases, which is reasonable because the tumor load is higher than oxygen, the oxidative stress is stronger, so increases, and then but the normal side, contralateral side also increases a little bit. So by this one, I had healthy the systematic source over here, but I also put a pathological source right over here. And then I try to increase the source intensity according to exponential growth of the tumor. So by doing that way, and then actually, so basically this one remains unchanged, but this pathological source increases in strength. By that way, the overall biophoton that you can observe on the surface increase at the right side, And then at this counterlateral side, it also can be increased a little bit. So this pattern actually agrees with that one. So these are the ways that I can use and try to model the trends. And again, the source biophotons, they can be linked to they need to be linked to the mind, consciousness. There is another one that's very interesting, that is potentially interesting, which is in 2016, there was a study published in PNAS, but it's very controversial, which they mentioned human high intelligence is involved in spectral redshift of biophoton activities. Well, which means that the different levels of consciousness or intelligence might change the spectral content of biophoton. And how that measurement was done with specimens, but something like this probably have never been observed in the live subject, but would this be possible? If this is possible, how we could explain that? While I would say that so this biophoton, whether it's organism, and then you're going to have different kinds of transitions to cause different spectral bands. And so a change in the relative intensity of the spectral bands will change the overall, will cause a red shift or blue shift. But in my model, I have this eventually when this one converts to light source, over here I have this transfer factor, actually they can be wavelength dependent. By doing that, by manipulating with this one, actually it's possible to model the difference of their spectrum, individual spectra, and the spectral content that could somehow correlate with different states, different levels of intelligence. And so, and then that will in principle, can lead to modeling the spectral shift. So overall, so what could this approach possibly lead to? I would say I would like to provide a path of quantitative means for correlating observations. So let's assume biophoton activities, it can, because of this oxidative stress, it can be correlated with our neuronal activities. But our neuronal activities, they will cause neurophysiological patterns, that are EEG patterns or some other patterns. It should be observable. So if they all come from this, they should have a path through neuroscience models and try to make a connection between these. But if that's the case, and then altered states of consciousness should also be kind of open to modeling, which will be very, very difficult. However, will there be a way to connect some kind of basically the strengths of their altered state of consciousness, how far away from the normal state of consciousness along this way, and how strong their observations that we can measure either biophoton or neurophysical patterns that is abnormal. And so can we make a correlation between them? And then we will have more tools, more information, more parameters to understand their phenomena that are difficult to do. And for doing that, I actually have another paper coming out in September in Physics Essays. I'm trying to use some additional ways of basically something related to modeling neuronal activity, not yet there, but how try to work within quantum mechanics, not bothering too much of quantum mechanics, and then try how to get there. And to summarize, and basically I projected some patterns of biophoton emissions of humans. They could be spatial, they could be temporal, they could be spectral. I consider that there might be a path to connect those patterns with our neuronal activity in the fundamental side. And then those, particularly the biophoton emission of humans at altered states of consciousness, even though we have a very limited data. And in order to reconcile those patterns, and might need to have a way to use some way to let neuronal modulation to regulate the photogenic process of biophoton emission. So make that path in order to connect between what we can measure from the object like biophotons, and then what we can actually correlate with the neuronal activity like EEG patterns. And I would like to point specifically to a study published in JSC in 2017, that the micro PK study in Brazil. And they mentioned that during the occurrence of a PK, there are substantial change, intense bursts of EEG patterns like that over here. So something like this, if you want to know more, I think the path is to try to implement ways, try to connect and have a neuroscience model to eventually bridge between EEG or neurophysiology patterns with the patterns that falls within the SSC and then parapsychology, this domain. So we can make a definitive correlation either from the intensity of the that we can measure, or from energy perspective, or from maybe entropy perspective. We ought to make that connection in order to deepen our understanding of those patterns that definitely will relate to consciousness, but what is the true relationship? So that's what all I have, and thank you very much.